Hello there and welcome to this Biology Mind A-Level tutorial on tests for lipids and proteins. In the last lesson we learned about tests for carbohydrates. We looked at the fact that there are some tests that are non-quantitative, there are tests that are semi-quantitative and quantitative tests. You now understand how to use Benedict's test, reagent test strips and iodine tests, and you understand that quantitative test examples include colorimetry and biosensors. A lot of words to get my mouth around that. All right. Okay, so we have three key learning objectives for today. So number one, the emulsion test. Number two, the Biuret test. And number three, chromatography. So let's kick off by thinking about the emulsion test for lipids. So what we do is we add ethanol to our sample of whatever we want to test and shake. We vigorously shake this mixture together for about a minute. And then we pour the solution into a test tube containing water. And we check for formation of a milky white emulsion. Now, this is the most key thing for you to remember here. So the emulsion test for lipids gives us a milky white emulsion. So that's our key finding of lipids being present. So why does this happen? Let's have a think about it. So lipids are insoluble in water. They will become immiscible if you try to mix them with water, so they just won't mix with water. However, they are soluble in ethanol. So any lipids in the sample form an emulsion. And the greater the concentration of lipids, the more of this white emulsion is formed and the more apparent the milky colour will be. So, for example, you can see here, this is a positive emulsion test for lipids. We've got this really nice milky white emulsion, remember, forming at the top of our test tube. So it floats to the top of the water, because remember, it's not soluble in water. So the lipids are not dissolved into this bit of water here. They've formed an emulsion with the ethanol that we've added. So that was our emulsion test for lipids. So now let's have a think about the Biuret test for proteins. So number one, what we do is we take our sample and we make the solution alkaline. So how do we do that? We add something alkaline to it, such as sodium hydroxide. We then add aqueous copper 2 sulfate to the sample. And if peptide bonds are present, because remember peptide bonds are the major bonds in proteins, then we get a colour change of our test solution. So if the solution remains blue, it means there is no protein present in the sample, and if the solution turns purple, there is protein. So purple, protein, blue, no protein. So again, this is just another result of an investigation that you just have to commit to memory, a bit like how you have to commit to memory that with the lipid test, with the emulsion test for lipids, we will get a milky white emulsion if lipids are present. So in the Biuret test, we're getting a nice purple solution if proteins are present. So now let's have a think about a specific technique we can use called chromatography. So chromatography is a technique which can separate many different types of biological molecules in a solution. Now this can be proteins, this can be carbohydrates, this can be vitamins, or it can be nucleic acids. Now the two types that are commonly studied are paper and thin layer chromatography. Now these two methods use the same principles. So what we do is we dissolve our test solution in a solvent. And we refer to this solvent as our mobile phase. So this is the phase that is moving, the medium that is moving in this experiment. Now typical solvents include ethanol and water. Now as well as the mobile phase, we have a stationary phase. And the stationary phase is usually a strip of chromatography paper, or it can be a thin sheet of glass, plastic or silica gel. When you do this in the classroom, what you'll be using is a sheet of chromatography paper. So our stationary phase is our paper, our mobile phase is our solvent. So just bear this in mind. Okay, so as the mobile phase moves through the stationary phase, the different molecules become separated at different points. So on this diagram, our stationary phase is our piece of paper, our mobile phase is this solvent in our beaker. Our solvent, or our mobile phase, is moving up our stationary phase. The solvent is moving up our paper. So you can see that this is the front of the solvent line on this piece of paper. You can see it's not quite reached up here yet, but it will with time. And you can see here that these molecules are becoming separated at different points. Now, this is because different molecules dissolve in the mobile phase to varying degrees. So there should be a clearly defined point of origin. So you can't quite see it on this piece of paper, but what we usually do is draw a line near the bottom of the piece of paper, and that's our point of origin, and that's the point at which we place our solvent to start travelling from. And that's also where we place a dot of our test solution. Okay, so that's our starting line. Think of it like a race. That's our starting line. So we can identify the different molecules using the RF value. So we determine the RF value by looking, first of all, at how far up our stationary phase the solvent has travelled versus how far our solute has travelled up the stationary phase. So there's a really useful little equation that you have to commit to memory. So it is RF value equals distance travelled by the solute over distance travelled by the solvent. Now remember, the solvent is our mobile phase, so that will be our ethanol or water, and the solute is whatever product we're testing, whatever molecule we're trying to separate. Alright, so this is an equation that you have to commit to memory. Now, essentially it's a ratio, isn't it? So the RF value is always going to be constant, as long as we keep the type of solvent used constant, so the mobile phase constant, as well as also the stationary phase, so the paper constant. And that gives you a constant RF value. So a certain solute or a certain molecule will correspond to a certain RF value. So we've already discussed this. So what we do is, first of all, we determine the distance travelled by the solute. So we measure from our point of origin, so our line on our piece of paper, to the centre of its spot. And that's the spot that's travelled up the piece of paper, up the stationary phase. And that's the distance travelled by the solute. And then the distance travelled by the solvent is how far the solvent has travelled up the stationary phase, i.e. the furthest point travelled by the mobile phase. So we measure the distance from the point of origin to the solvent front on our stationary phase. And then we plug those two measurements into our equation to calculate the RF and compare this RF value to a set of known RF values, and that's how we find out which RF value we get in our experiment corresponds to a definitive RF value of a molecule. So just to show you, you're going to plug one number into here, one number into here, and you're going to get your RF value. Alright, so that's lesson 18 completed for today. I'd say from this tutorial, I think the main thing for you to take away is the results of the lipid test. So what forms a positive lipid test? It's a milky white emulsion. Similarly for the Biuret test, you need to know what happens when a protein is present in the solution. And lastly, understand this concept of the RF values in chromatography. Alright, well done for today, and I will see you for the next tutorial.